My name's Juliet Graham, a paediatric oncologist from Southampton, and I'd like to welcome you to the third session, which is continuing the theme of relapse and refractory disease. And I'm delighted to say we've got two really excellent speakers joining us today. Dr. Lynley Marshall, who's a consultant paediatric and adolescent oncologist from the Royal Marsden Hospital in Sutton, and Dr. Steve DuBois, who's a paediatric oncologist from Boston Children's Hospital. And they are going to give us insights into options for relapse and refractory disease on both sides of the Atlantic. To start off with, I'd like to welcome Dr. Lynley Marshall, who's going to give an overview of early phase trials in Europe. Thank you, Lynley.
Good afternoon, everybody. It's my pleasure to be here speaking in this session and to see so many people here. Here we go. It is an overview of trials in Europe and in the UK, and it is a little bit of a flavour of what we have rather than a comprehensive overview. We don't have the time for comprehensive overviews here. The takeaway messages are that the focus of our early phase trials must always be on the impact it makes for patients and families, that international collaboration and partnership is absolutely essential in making faster progress, and that trial designs need to be seamless and efficient. And platform trials may be one way that we can achieve that, in part at least, so that they can adapt as the science evolves. The goals of our cancer drug development for our patients and families must be to try and use precision medicine and personalised approaches where possible. And as we've heard from Ro this morning, of course, that isn't always possible in neuroblastoma, particularly frontline. We need to have studies that are cross-tumour, but then panning out into disease-specific studies when we can. To have cross-age studies so that they are inclusive and don't deny access to patients based on arbitrary age barriers. And of course, we want to undertake bench-to-bedside and back-again approaches where we're learning from every patient that's enrolled in a clinical trial. We want to get the right drug to the right patient at the right time in their cancer journey, and ideally move the best drugs to more frontline treatment more quickly. And as we've said, we want to reduce toxicity and improve the quality of survival as well, notwithstanding some of the toxicities we've seen with the CAR-T studies, and striking that balance, of course, is what we need. But the studies must be patient-focused and very much led by the patient and parent voice. In terms of impact for patients, we want accessibility to clinical trials and novel therapies. The trials are the treatment for many patients, and so lack of access can impact outcomes. At our centre, and many that you will be treated at, we have an ethos of equitable access where possible, and we know, unfortunately, that it isn't always possible. But we try wherever possible that every newly diagnosed patient with cancer in the paediatric or TYA age range and at every stage of refractory or progressive or relapsed disease is at least considered for available clinical trials where they are there, and for additional molecular profiling if that's thought likely to be potentially helpful. We've spoken a lot about what early phase trials are. Just to level the ground quite clearly, phase 1 trials, including first-in-man trials and first-in-child trials, test new therapies, and sometimes they are in combination in a relatively small group of patients to avoid overexposing too many to potentially less effective treatments. They usually are in patients who have had other lines of treatment, and they are often across tumor types in the first instance, with expansion cohorts sometimes in relevant diseases where a certain target may be relevant. And it aims to study the best dose, the best pharmacokinetics, i.e. the levels of drug in the blood that are needed to achieve the responses we're looking for, signs of toxicity and tolerability, and of course, early signs of anti-cancer activity. Phase 2 trials historically test the treatment in slightly larger groups of patients. They are often disease-specific, such as in neuroblastoma. They are more likely to be randomized. They allow refinement of the doses and the tolerabilities and toxicities that we learn about at the earlier stage, and there's a greater focus on anti-cancer activity and efficacy. But increasingly, these are artificial divisions, and most studies these days are seamless phase 1-2 studies where they roll into each other, and that saves time and helps us to bring things forward more quickly. And of course, even in phase 1, it's important to have a line of sight to where a drug, if proven useful, could be utilised at an earlier stage in a relevant disease type. And that's why working with the cooperative tumour groups is really important. And of course, the phase 3 trials are the upfront treatment trials, usually in newly diagnosed patients, standard of care trials, but they may include novel agents that have come through the earlier lines of testing. I think it's important to mention this again, and we touched on a couple of them in the morning session. There always has to be potential for individual patient benefit, even in early phase trials. The starting dose is usually extrapolated from adults. It's very different to an adult phase 1 study where they literally are picking very, very tiny doses to do no harm, but much less likelihood of benefit too. It's also important that we don't use placebo-only trials in pediatric cancer. That sounds obvious, but it's important to be clear about that. You know, everybody in a pediatric oncology study gets active treatment. Of course, we do have studies where there is a backbone with a novel agent or not, and in that case, sometimes there are crossover designs. There are, by necessity, strict eligibility criteria. We try to be a bit permissive where we can be, and we know blood tests in neuroblastoma is a particular issue where we have to have disease-specific parameters rather than the same standard as other studies because of bone marrow involvement in many patients. But we do need to have good organ function, good so-called performance status, so that we do the studies safely, we get good high-quality data, so that if they are positive, they can be hopefully moving forward to even authorization and benefit many, many more patients internationally. It needs close monitoring, and that's important so that you can pick up early signs of new toxicities emerging. But again, as mentioned earlier, decentralized approaches where that is possible, so you're not, flying hundreds of miles for every blood test, is really important, and an increasing focus in the international community. Development of age-appropriate formulations is also something that we consider in the kind of phase 1-2 world, looking at dosing, taste, palatability, and whether they can go down NG or NJ tubes. And of course, we have to work very closely in partnership and communicating with all teams involved, referring centres, community, GPs, and of course the shared care centres within the UK setting. And this is key so that we are picking up the early toxicities in a timely way and can report them in real time. I think it's also important to stress the parallel approaches that are necessary, working closely with symptom control teams, and that's twofold because the symptoms may be disease-related or they may be related to the new therapy. And if we can best manage symptoms such as pain, nausea, vomiting, whatever they may be, that gives a much higher likelihood of staying on the new therapy for longer and getting benefit and hopefully getting these drugs forward. As was said earlier, neuroblastoma is rare. It's not rare in this world, in this room, or indeed in our world, but it is rare. And so the best way to work together is collaboratively. We work in the UK setting across our 20 tertiary centres in the TCLG, and many of you would have been treated there. The smaller early phase network, the ECMC, is an adult and paediatric all-age network, but comprised of 12 paediatric centres that focus on phase 1-2 studies, and then 5 ITCC designated first-in-child study centres. And within Europe, we work across the ITCC network and with the SIAP Europe disease-specific group, SIAPEN, and then more broadly internationally through the Accelerate multi-stakeholder platform, and in the neuroblastoma group, the NAND consortium, and COG. And in the ITCC, this is a European network of 62 paediatric oncology centres, many focused on neuroblastoma and other early phase trials, 25 research labs, again many focused on neuroblastoma, with memoranda of understanding beyond Europe. And we know that our pharma partners have global development plans, so very often these first-in-child studies are being run across multiple countries, and when we're all on our safety calls, there are people up at all hours of the night to join them. Prior to the Beacon neuroblastoma trial, I think it's important to say there was never a standard treatment for relapse, and that is the context that this trial was initially set up, designed by Lucas Moreno in collaboration with Professor Andy Pearson, who's in the room. And this is a summary of the study, but sets the scene for what comes next. So Beacon which is now made up of Beacon 1 and Beacon Immuno, recruited 225 patients across 10 countries over 8 years. And essentially, it was what was called a factorial design, whereby in Beacon 1, you test the best chemotherapy backbone to which novel agents can be added so that patients weren't having serial phase 1 study. And in this situation, it was the monoclonal antibody, anti-VEGF antibody, bevacizumab, that was being tested. As that was coming to a close, it was converted to Beacon Immuno, and the chemo backbone was being tested with the addition or not of dinutuximab beta. Beacon, of course, has been presented and being published at the moment, but these were the best arms of the two parts of Beacon. So BIT with a response rate of 30% overall, or 30 out of every 100 patients responded in the part 1, and the dinutuximab beta with a response rate of 35 as the best arm in Beacon Immuno. So the summary was that Beacon identified 2 regimens with signals of activity in a randomized setting. And the chemotherapy backbone, there was no difference whether you used irinotecan or topotecan with temozolomide, but it was clear that temozolomide alone was not a good idea. And we also learned that refractory and relapsed disease behave differently to each other. So Beacon 2, which has opened recently in the UK, sponsored by Birmingham and led by Lucas and Juliet and others, will compare and confirm the efficacy of the established combinations through Beacon 1 and Beacon Immuno, and also test new combinations. And this is a real opportunity. So this is open in the UK and it's rolling out across Europe, but the design is such that there's a tier 1 and a tier 2. Tier 1 is for patients at first relapse of neuroblastoma only, and will test the best 2 arms out of the original Beacon. So BIT versus dinutuximab beta BIT in a staged way with planned analyses to go or no go and keep expanding. Tier 2 is a different approach that will have multiple different smaller arms testing novel agents. The first of that is arm C, and as you can see, that will test the combination of both dinutuximab beta and bevacizumab with irinotecan and temozolomide, but only in 10 patients to get an initial signal. Is that tolerable? Is it too toxic? Putting both of the antibodies together. And then additional arms will be added in, arm D, arm E, etc. And it acts as a platform through which new agents can go in. And these are just some of the considerations that have been under discussion through the NDD group. You can see there's immunotherapy approaches, there's a copper chelator, which is likely to be the next arm that goes in from work done out of Australia. There are drugs that we've tested in earlier studies, such as BCT100 was tested in the PARC study. That could potentially go in combination. And there are anti-PD-1s, CDK4/6s, TIGITs. But the key thing is that it's adaptable and very much open to new ideas from investigators who have them and can lead on these arms within the study. Another phase 2 study that is open in Europe, and it's open in Leeds in the UK and elsewhere. We heard earlier the naxitamab study. So naxitamab is a humanized monoclonal IgG1 antibody which binds to GD2. This is a study which combines it with GM-CSF. It is for refractory disease rather than for relapsed disease, and it is quite a toxic treatment. As we heard in the earlier part of the day, you really need to be in a specialist centre. The levels of hypotension and pain are really quite eye-watering, but the recovery from that is quite rapid. So just highlighting that courtesy of Martin Elliott in Leeds. Coming on to the phase 1, the truly earlier stages, we know that the precision medicine approaches around Europe, such as stratified medicine PEDS in the UK, but MAPIACT in France and elsewhere, ITHER, Inform in Germany, has really caused a paradigm change where we've moved more likely to biopsy at the time of relapse to see if any new actionable aberrations have appeared in the tumour. And we know that this can happen and can be a mechanism of disease resistance. So SMPEDS profiled 806 UK patients from around the UK, CCLG centres over an almost 5-year period. Lou Chesler was the chief investigator, and Sally George has been heavily involved. Multi-omic profiling, and depending on that profiling, patients could be put onto either authorised drugs if you were fortunate enough that there was a drug authorised for whatever aberration you found, and there were a few, or clinical trials being run by drug pharma partners or other academic trials, including eSmart, which is our European platform study testing novel agents, novel combinations in a phase 1/phase 1b all-comer setting. Just to say as an aside as well that StratMed Peds 2 has just launched in November 2024. Again, Louis is the lead on that, and Aditi Vedi has been heavily involved in writing the protocol. That is rolling out across the UK, and that will test rapid turnaround technologies such as nanopore to be validated in children, but also much greater use of liquid biopsy so that one day there may be a setting where we don't need to do the tumour biopsies to get the answers we need and to triage into trials. So a very important study. This is a summary of eSmart, and it's not there to make you look at every single arm, but it's to show, led by Birgit Gerger out of IGR, different arms of novel agents and combinations of major interest in pediatric cancers previously, but not accessible. 6 pharma partners have put their drugs into the study. You can see the arms that are marked in red have completed recruitment or close to recruitment or met their go, no-go decision as per the protocol. Other arms are still open, and it is an adaptable study where new arms can go in. So it's a cross-disease study, but of course some of those agents are of interest in neuroblastoma. The fadraciclib CDK2/9 inhibitor worked up in Lou's lab and licensed out to Cyclacel of major interest preclinically. The adult study paused to change the formulation from an IV to an oral tablet, and so that's been halted, but we very much hope we would have access to CDK2/9 inhibitors because they are of major interest in the disease. Just a mention of the CRUK-sponsored Determine study. This is a UK-only study. It's a paradigm of testing licensed drugs in unlicensed indications, particularly rare cancers. By definition, pediatric cancers are rare, but it's also adult rare cancers, TOA rare cancers, or common cancers with rare genetic alterations. Again, a platform design whereby molecular profiling, patients are put into the study, allocated to an arm based on genomic matching, followed up looking at best disease control rate over a 6-month period. But the key thing is that we have agreement with NICE and with NHS England that if there are positive arms, they will be looked at by the Cancer Drugs Fund for authorization and for reimbursement of those agents for patients with rare cancers. And so that's a new paradigm as well. The current status is that Roche and Novartis have put drugs into the study, and there are others that I can't yet announce, but there are agreements in place, and those in purple are of some interest in neuroblastoma. We know with other ALK inhibitor studies that have closed, access isn't always so easy, but the alectinib is in there as an arm in the study, and atezolizumab may also be of relevance for some neuroblastoma patients. Just mention of other phase 1/2 trials of relevance to neuroblastoma. So the ALK inhibitor studies, we know, up to, depending what testing you use, 10 to 15% of patients at diagnosis may have ALK aberrations, but we know that's more common at relapse and disease progression. So the lorlatinib study was a NAD-sponsored study run out of the US, but with a centre open in France at Curie, which Gudrun led, and at the Royal Marsden, and that's been really instructive for being a paradigm for ALK inhibitors and moving them more forward, and I'll just talk about that in a bit more detail. That's close to recruitment, but we actually still have patients benefiting from the drug who are receiving it now outside of the study. The CRISP study is a multi-tumour study, but for neuroblastoma it combines crizotinib with the mTOR inhibitor temsirolimus. That arm has just closed, and we'll be looking at the data for that. And then there is a Roche standalone alectinib study, again, which is for ALK fusion rather than ALK mutated tumours, and so relevant for a very small minority of neuroblastoma that have the fusion of ALK rather than a mutation. And then I've mentioned Determine. So combination strategies, first and second generation ALK inhibitors, with a view to moving them forward. So this was what the CRISP arm was, crizotinib with temsirolimus. We know crizotinib is authorized already for 2 other tumor types, anaplastic large cell lymphoma and inflammatory myofibroblastic tumors. We know that much higher doses are needed in neuroblastoma, and also Pfizer is keen to collect additional information around eye toxicities, and so we're doing that through this study. Lorlatinib, a really important study, successful. Kelly Goldberg, who's here and speaking later in this conference, was the first author on the paper, established the recommended phase 2 dose of lorlatinib with and without chemotherapy, and you can see them there, but very good response rates, particularly in the TYA population over 18, even as single agents, and so that gives very good evidence for taking that forward, even in a frontline setting, and there are initiatives ongoing to demonstrate benefit there. Phase 1-2 studies with CDK4/6 inhibitors, MDM2 inhibitors, others. We've had a raft of studies with CDK4/6 that have been running, and actually very recently just closed to recruitment, some recruiting up to 21 or 25 years of age. The paradigm of a backbone chemotherapy with an added targeted agents such as ribociclib or palbociclib, both of those expanding in neuroblastoma because felt to be a good biological rationale. The idasanutlin study, very similarly, those studies will publish, but one of the key challenges that has become clear from them is that finding that Goldilocks sweet spot of where you can actually get a high enough dose in patients that have been very heavily pre-treated, as these patients would have been. It's not usually first relapse, it's patients that have had multiple other diseases. We're learning from those patients as we go along. And so the dosing challenges in later stages of neuroblastoma present real issues to try and fairly judge these agents, and judging them earlier on would be far preferable where possible. We've got the PD-1 and DNA damage repair inhibitors. So the pembrolizumab study from Merck is still available for patients that have a high tumor mutational burden or MSI high. We do see some neuroblastomas with that. The GSK study of the PARP inhibitor niraparib with dostarlimab is ongoing at the moment and expanding in neuroblastoma and osteosarcoma. And then the olaparib study actually has just closed this week. Single agent study, lack of feasibility. They couldn't recruit enough patients in the whole world with all diseases, and so that's a shame. But the good thing is that we have PARP inhibitor studies in combination. There were combinations in eSMART, and Sally George is leading an MIBG molecular radiotherapy study of MIBG with talazoparib. So SCOOP, this is the niraparib dostarlimab. Part 1 testing the niraparib tablet with the immune checkpoint inhibitor. Part 1B is testing a tablet that can be suspended for younger children. And then, as I said, safety run-ins, but expansions in neuroblastoma and osteosarcoma. Sally's MINT study in very late stages, just about to go to regulatory, absolute labour of love in collaboration with our transatlantic colleagues. And Daniel Morgenstern's been involved in that, and others who are here. So this is looking at a biomarker-driven trial and looking basically difficult to do a randomized study in this setting, but at least have data from a relatively recent NANT 1101 study, which is the largest contemporaneous MIBG study that can act as a close historical comparator. And this is combining the PARP inhibitor with MIBG over a short period. Credit to Sally for that. So that's just a flavour of the types of studies that we have in the UK and Europe at first relapse and beyond. Takeaway messages: the focus must be for patients and families, not just research for the sake of research. And we really do take the approach of trying to learn from every patient. When we're on safety calls, we're discussing individual cases, and all the investigators on the call learn, obviously not by patient's name, but really helpful to learn from the experience of others. We have to collaborate to move forward more quickly, and part of that collaboration and partnership is very much with patients and families. In our academic studies, they sit on our trial steering committees. They help us design the studies. They are the golden thread that runs through all of our research and makes it better and faster. And then finally, trial designs need to be seamless and efficient. We shouldn't have gaps between trials wherever possible. And so even now, I'm sure somebody's thinking about Beacon 3. I'm not sure who's going to take that on. Thank you for your attention. Just to acknowledge all our patients and families, all our smaller patients and family charities, as well as the big players who fund research, and to everybody who's here today. Thanks very much.
So thanks to Lynley for a really excellent talk and start to the session. I'd now like to invite Steve DuBois to come up and to give us an overview of what's happening in North America. Thank you, Steve.
Thank you, Juliet. Really appreciate the introduction, and thank you to SKC for hosting this event and for inviting me to speak. Really honored to be part of today's session. And thank you to Lynley, who set me up actually perfectly, because I think we have nearly non-overlapping content for the parents. So that's very nice. So I'll give the North American perspective on novel therapies for children with relapsed neuroblastoma. And I'm at Dana-Farber and Boston Children's, where I lead our early phase clinical trials effort. These are my disclosures. And so what I thought I would do over the next 30 minutes or so is to give an overview of really who the players are in North America. You'll hear some of these names, but it's just helpful to sort of lay that out, but also talk a little bit about how those clinical trial sponsors interact to try to ultimately advance novel agents into the frontline space. Then I think it'd be helpful for this audience to hear some of the challenges that we face as people developing new drugs for children with cancer more broadly and neuroblastoma specifically, with hopefully a plug to charge you all with becoming new advocates for the field, because we certainly need the patient family voice to get access to novel therapies for our patients. And then I'll, at the end, provide some of our recent, current, and planned trials for children with relapsed and refractory neuroblastoma. So first, who are the people who are sponsoring clinical trials in North America? And so we have a few networks that are involved here. So there's the NANT New Approaches to Neuroblastoma Therapy Consortium that's based out of Children's Hospital of Los Angeles, and they have traditionally sponsored clinical trials for patients with multiply recurrent disease. There's the Beat Childhood Cancer, or BCC, led by Dr. Scholler, who many of you have mentioned, out of now Hershey, Pennsylvania. And then, of course, the Children's Oncology Group, and I'll talk about how that fits in here as well. And then, of course, there's any number of individual academic sponsors who might sponsor trials. So Dana-Farber, where I practice, or Memorial Sloan Kettering. Those would be individual academic hospitals who are sponsoring, more commonly, a single institution study. And then, of course, our industry partners are also sponsoring clinical trials, more of those being transatlantic, which I think is a reflection of the resources that the pharmaceutical industry has to lead those types of trials. So our usual paradigm is shown here for developing a novel agent, and it typically starts with testing something that shows interest in usually the laboratory, and testing that in a population of children with multiply recurrent disease. Those trials are usually done by the NANT or BCC, the individual industry sponsors or individual academic sponsors. And then if something is really showing activity in that population, the goal is to have that move forward into a first recurrent randomized trial to really demonstrate that there's real benefit of adding the novel agent to standard therapies. And given the size of those trials, those first relapse trials in North America are typically sponsored by the COG. And then, of course, if things are really looking good in that context, then graduating even further to a frontline randomized trial. And those are typically many hundreds of patients with newly diagnosed high-risk disease. And so really the only group that can sponsor a trial of that size in North America is our COG group. And of course, what we would love is for there to be something that shows such striking activity in one of these early trials to immediately graduate to the frontline space. And we have done that once with the lorlatinib experience that Lynley shared, where that NANT clinical trial helped to inform actually a trial that we're doing in North America for children with alkebarent high-risk disease, and a forthcoming modification to HR2 here in Europe and the UK. So with that background, let's maybe look a little bit more critically at how our field is doing in terms of developing new agents for our patients. And so I almost can't give a presentation without showing this slide. It's an analysis I'm really proud of, led by a medical student who cataloged 117 FDA-approved cancer medicines, so not drugs that never made it to market because of toxicity or lack of efficacy, but real medicines, so 117. And this medical student cataloged the time from the first in human clinical trial to the time to the start of the first in child clinical trial. And what we showed was that time lag was 6.5 years, which is, I think we would all agree, unacceptably long, and something our field is really grappling with and trying very hard to reduce that time lag so that we get promising agents into the pediatric clinic much more quickly. Another way of looking at this is to look at a large number of what we call new molecular entities, basically drugs, whether they're approved or not, and track what happens to them a decade on. And so that's this type of analysis that we've conducted. And so what it shows here is the cumulative probability from the start of clinical development, usually from the time that new drug enters the adult clinic. And if you sort of track that forward, you can see a couple of things that are a bit sobering. Well, actually, everything on this curve is sobering, and I'll take you through it. So first, the probability of any of these drugs getting an FDA approval is shown here in the blue, so 13.9%. It just tells us, cancer drug development is really hard, right? And so we all want new medicines to be, to be on market and available for patients. But even in adult cancers, where obviously there's a larger patient population to recruit to clinical trials and to hasten development, the likelihood of an approval remains low. The other sobering point is, of course, this sad little green curve down here is the probability of a pediatric approval. So 10 years from initial start of clinical development, 1.4%. So really hard to get a pediatric cancer drug approved. And then the other curve here, this orange curve, is the cumulative probability that the drug would enter a pediatric clinical trial. So of all of the drugs that enter the clinic, only about 23% are entering the pediatric clinic. So you can tell we're sort of swimming upstream here as pediatric cancer drug developers trying to get these novel agents to your clinics. The other important metric, I think, is how commonly our industry partners are sponsoring clinical trials. So this was another analysis that we did using publicly available data from clinicaltrials.gov, where we looked at the percent of clinical trials that are sponsored by industry in black or non-industry in blue, and we stratified that analysis by age. So we looked at all ages, age of eligibility under 18, and age of eligibility greater than or equal to 18. And so what you can see here on this panel, A, for the non-oncology trials, is you can see that about 40% of trials are sponsored by the pharmaceutical industry. And you can see, if you look at the pediatric trials, there's a small fall off here compared to adult clinical trials. If you look only at oncology trials, though, you can see that fall off is much greater, and there's fewer pharmaceutical sponsors of pediatric oncology clinical trials. That's important because in data I'm not showing here, the trials that were sponsored by industry were able to enroll more patients and to do so more quickly. And so if our collective goal of those in the room is to try to get newer therapies to our patients more quickly, it seems as though there's an advantage for the industry trials, but those are underrepresented in pediatric cancer. We also have historically relied very heavily on monotherapy trials, and we have a big push internationally to try to improve upon this. But these are some data that we put together using, again, publicly available data from clinicaltrials.gov, where we looked at all pediatric oncology phase 1 clinical trials, and we looked at the types of therapies that were being tested in those trials. And so what I show here, this is again the proportion of trials, they all sum to 100. The orange are the monotherapy trials, and the blue are combination trials. And so you can see for phase 1 clinical trials that only enrolled children, so patients under 18 only, that most of these clinical trials were monotherapy trials. And as the age of eligibility increased, the probability of being a combination trial really increased. And we think, historically, most of our benefit in pediatric oncology, not just in neuroblastoma, has really come from combination strategies. So we've put together, under the leadership of Dr. Pearson, a position paper about really informing the field of what we think is really best practice for doing this. And I think I'm encouraged by actually the slides that Lynley showed, where most of those trials that she reviewed really were combination approaches. So I think this is changing for the better. So why am I telling you all of this, right? This may not seem immediately applicable to your own child, but I think is incredibly important for you when you are feeling frustrated. We're feeling frustrated, right? And we really are. We are really working very hard to try to get these agents that we really think may be beneficial into the clinic, and to do so more quickly and in combination. But the real reason I'm telling you this is that really we need your help. When we go and advocate as physicians and researchers, that's not as powerful as when the patient voice is present. So it would be fantastic if an outcome of this presentation is that more of you take on a greater advocacy role, perhaps with SKC. Yeah. How's that for a plug, Leona? Is that good? Thumbs up from Leona. All right. With that, I'll just share some of the clinical trials that we've been interested in fairly recently in North America, and I'll divide those into 4 bins: chemoimmunotherapy, CAR T cells. I'll spend one slide on because of the really nice session we had this morning. Radiopharmaceuticals, and then the antibody drug conjugates, which are really, I think, increasing in interest. So first, the chemoimmunotherapy, I think to say, certainly from the Beacon Immuno trial and from our own trial in the COG, ANBL 1221, has really taught us that the combination of a GD2 antibody like dinutuximab or dinutuximab beta together with chemotherapy really has led to a very high response rate. About 50% of patients will respond to that combination. And so now the real push has been for our field to try to build upon that success and try to understand what agents may we combine with chemoimmunotherapy to improve its activity. I'm going to take you through 5 slides of the North American trials that are really, or North American sponsored trials that are trying to do just that. So the first trial is open and enrolling and is sponsored by the NANT Consortium, and the innovation here is to administer natural killer cells that really play a key role in helping GD2 antibodies work, is really to administer those cells alongside the chemoimmunotherapy regimen. So that's one option to really try to boost the effect of chemoimmunotherapy. The second approach that's our colleague Dr. Goldsmith is leading at her center in Atlanta is to give a different type of cellular product, gamma delta T cells, alongside the chemoimmunotherapy. And it's the same idea. It's really trying to increase the immune effects against neuroblastoma when you give a chemoimmunotherapy strategy. With these gamma delta T cells, though, it doesn't require the effect of these cells don't require GD2, and so sort of thought to be 2 different approaches to try to boost the immune system. So this is a trial that is also open and enrolling. Then we have a few trials in development. Our colleagues at St. Jude are collaborating with the NANT to develop a clinical trial of chemoimmunotherapy alongside a drug called IL-15 or interleukin-15. And this drug is thought to both increase the number and function of these natural killer cells, again, trying to boost that immune response from this combination. So this is a trial still in development. Another NANT trial will give a drug called a TIGIT inhibitor alongside chemoimmunotherapy, and this is a nice collaboration with a laboratory at the Princess Maxima that showed that this TIGIT is an immune checkpoint that can reduce the response to immunotherapy. So if you can inhibit the inhibitor, then you can boost the immune response. So this will be a trial sponsored by the NANT, but will ultimately be open here here in, at least in Europe. I don't know actually about the UK, but this remains in development. And then just last evening, we received the good news that this trial was approved to proceed. So this will be a children's oncology group clinical trial of a drug called iberdomide alongside chemoimmunotherapy. And so we've just been approved, and so very early in development, but this will be specifically a trial for children with first recurrent or first refractory high-risk disease. And so this iberdomide drug likewise stimulates the number and function of NK cells, and this will be a randomized trial so we can isolate the effect and really determine the role of iberdomide in that patient population. So If it sounds like I've been repeating myself, I have, because we've now got five trials, all using more or less a very similar approach of chemoimmunotherapy and trying to build upon the efficacy of chemoimmunotherapy. And so, if we have sort of a big picture view, that's exciting that there's a lot of work in this space, but it's a little bit worrying as well that a lot of our eggs are in the chemoimmunotherapy basket at the moment. I think appropriately so, but something that I think we all have to maybe keep an eye on over time. So I'll transition just to show one slide, maybe with some animation, I think. Yes, with some animation on CAR T cells, and I'm not going to belabor the point because of the really wonderful presentations that we had earlier today. But we heard a lot about GD2 targeted CAR T cells, and we have these approaches in North America open at several centers. But we also have other targets of interest. So we, for example, have just activated a trial of an ALK CAR for patients both with and without an ALK alteration, just leveraging the overexpression of ALK on neuroblastoma regardless of mutation status. There are several centers who are developing B7H3 CAR T cells, and then our colleagues at the Children's Hospital of Philadelphia has a trial targeting GPC2. So there's a lot of space and a lot of interest, a lot of interest in this space in North America as well. One of my real interests has been in radiopharmaceuticals, specifically MIBG therapy. So I thought I might spend just a moment orienting people to really what we're talking about when we're talking about radiopharmaceuticals, or as Mark likes to call it, molecular radiotherapy. Is that right, Mark? Yes. All right. And Mark's just published a really nice review article in the Journal of Nuclear Medicine that sort of sets the stage. So if you're interested in learning more, that's a great resource. But really, what we're talking about here is similar to CAR T, leveraging a cell surface marker to tag in a radioisotope that then will damage the DNA in the neuroblastoma cell and lead to death of that neuroblastoma cell. And so when we've been talking about MIBG therapy, that's taken up via the norepinephrine transporter, but there's other targets of molecular radiotherapy that are relevant to neuroblastoma, including the somatostatin receptor with dotatate, and then the GD2, which we've been talking a lot about. You can tag an antibody to GD2 with a radioisotope and achieve the same effect. So I'll just give you a little bit of a flavor of what we've been working on in the molecular radiotherapy space. And so what's really nice about this is that you can select a patient for these types of therapies with an imaging approach, right? And you're not using tumor material collected 3 years ago. You're doing a scan like a few weeks before you're going to treat the patient. And so we call this a theranostics approach, where you do, in this case, an MIBG scan, and if you see multiple sites of disease, that renders that patient a candidate for molecular radiotherapy using I-131 MIBG. And likewise, if there's no evidence of MIBG avid disease, as shown here, then that patient would not be a good candidate for molecular radiotherapy, at least for that target. You could certainly do a dotatate scan on that patient to see if they might qualify for a different targeted radiotherapy. So Lynley had mentioned the NANT 1101 trial. This was a trial that I had the opportunity to lead through the NANT consortium for children with recurrent MIBG avid neuroblastoma who were enrolled and randomized at study entry, and they were randomized to receive MIBG monotherapy on arm A. On arm B, MIBG together with 2 chemotherapies that we thought might make the MIBG work better, and then MIBG together with an oral inhibitor, a targeted therapy called vorinostat, that we likewise thought might make the MIBG work better, but we didn't know, and really we felt the only way to really know was through a randomized trial. And so at the conclusion of the trial, we were able to demonstrate that the children who were randomized to arm C with MIBG plus vorinostat had a higher response rate compared to the patients on the other two arms of the trial. In addition to those objective responses, there were a number of children on each of the arms of the trial who had what we call minor responses, where at least one part of the disease improved, even if the other parts of the disease did not improve. So the successor to that trial has really been, in this era, not to look at chemoimmunotherapy, but radioimmunotherapy, where we're combining a GD2 antibody together with MIBG therapy. So this was led by our colleague, Dr. Thomas Cash in Atlanta, and was the NANT 1701 trial, where we combined MIBG together with dinutuximab and showed a response rate of 42%. At the time that trial was kind of wrapping up, the NANT 1101 results came out showing the improvement with vorinostat, and so it was a natural extension to add vorinostat to that to test in Part B this 3-drug combination of MIBG, dinutuximab, and vorinostat. And we showed a response rate of 42%, but there were a number of patients as well who had minor responses on Part B of that clinical trial. So at this point, we really find ourselves in a very similar situation where we don't know if adding the dinutuximab to MIBG is better, adding the two drugs, the dinutuximab and vorinostat is better, or just the original combination of MIBG and vorinostat. And so our colleague, Dr. Vo at UCSF, will be leading this trial that we expect to open later this year, and will be very similar to the NANT 1101, where patients will be randomized to one of these 3 arms. And gratifyingly, no monotherapy on this trial, so everyone will get some form of a combination. There's some preclinical work looking at a radiopharmaceutical directed at GD2. This is something called a SADA approach, and these are the results of a mouse experiment where on this axis you see the tumor volume, and then this is time after the treatment. And you can see with the GD2 SADA really sustained reduction in tumor volume compared to if you just give the non-targeted radioisotope alone, those tumors in the mice just increase in size. And not surprisingly then, if you look at a survival curve in the mice, all of the mice had prolonged survival when treated with the GD2 SADA. So this is a clinical trial that's now open for patients 16 and older, and patients with neuroblastoma are eligible for that. But really, to properly test this approach in patients with neuroblastoma, we need a dedicated pediatric trial, and so we're partnering with YMABS on this study that will be a collaboration with the NANT. And then just to say, for the somatostatin receptor targeting in North America, we have, I think, one open trial using copper-67 sartrate that is targeted to the somatostatin receptor. And so that's open and enrolling. It is an industry-sponsored trial. So let's talk then about antibody-drug conjugates, because we're hearing more and more about those as time goes on, and it's not dissimilar to what I've already shared with you, where we're really leveraging cell surface targets to bring in a payload. And so those targets might be something called ROR1 or GD2 or B7H3, and so really each of these targets are bringing in a low dose of chemotherapy targeted to the tumor cell, potentially sparing children the effects of chemotherapy on normal tissues. And so there are several of these either already open or soon to open that I think are really of great interest for children with relapsed neuroblastoma. And then there's the potpourri that didn't fit on the other slides, but there's a wide range of other things that we're interested in with a range of different sponsors. And as I put this slide together, I was also really happy that nearly all of the children who will participate in these clinical trials will receive a combination. In some of them, there's a small portion who need to get the single agent so that we can learn a little bit about the drug in children before we then use it in combination. But for the most part, these are combination strategies, which I think is very gratifying. So if I were to summarize my comments, I would say, hopefully you've heard about some of the challenges that we face in pediatric cancer drug development, and that you're feeling a little fired up and talking with your, what do we say, a member of parliament? Talk with your members of parliament about that. But despite all of those challenges, we have a lot of drug development activity at the moment, with a big emphasis on chemoimmunotherapy, with 5 trials either open or in development. Obviously, the CAR T-cell remains a very active space, radiopharmaceuticals, and the antibody drug conjugates. And I think collectively, we all really need to advocate to access the best drugs more quickly, and then once we have permission to study those drugs and access to those drugs, we really have to design the best trials for our patients, which in my view means combinations. So I'll first and foremost acknowledge the patients and families who have the courage to participate in our clinical trials, the collaborators on a lot of my work, our trainees who did generated a lot of the data that I shared about our drug development metrics, industry partners, and of course, our funding sources. So thank you.
Thank you, Steve. That was a really excellent talk. And Lynley and Steve have both stuck beautifully to time, so we've got lots and lots of time for discussion, which is great. So I'd like to invite Nick Bird and Stuart Lever to also come and take a seat on the panel so we can open up for discussions. Sorry, Lynley, do you want to come up too? And so maybe actually while people are just thinking of questions, I can start with a question for Lynley. Actually, that you had a very long list of different trials and studies, some of which are open, closed, may not be open in different centres. How would a paediatric oncologist in a centre that doesn't necessarily run those trials know that those were happening and potentially offer them to a patient in the UK?
So we have a very tight network within the UK. Through the ECMC network, there is a website called EC Trial Finder. It is for professionals in pediatric and adult oncology who can look for the disease type it in and see where the trials are, gives information about where the study's open, who the chief investigator is. There are studies on the CRUK website, for example, and also now going back onto the CCLG website, we're going to be having early phase study newsletter again, which will be available for families as well. But it's often, investigator to investigator. If, for parents, always go to your child's oncologist, and if you know about a trial or if you want to know about other trials, most of us are very happy to refer. We have our ECMC regional relapse network, so across 4 different regions grouped geographically. We have the regional relapse telephone conferences, 1 to 2 weekly, where 4 or 5 centres will be discussing all patients within that region, not by name, but presented by the oncologist, who either have molecular profiling data available to discuss in that setting whether there's an early phase trial, or indeed to discuss trial eligibility and molecular profiling. And the StratMed Peds 2 molecular tumour boards will be starting up again soon now that that has opened, and that's another forum where oncologists can dial in and listen to what's available for their patients.
Great. So
And of course, the SKC
Yeah.
team is incredibly well informed about the available trials, so,
And directing patients to the centres where they're open, of course.
Sure. Thanks, Lynley. Question from the audience. Carrie, just wait 2 seconds for a microphone. Just while we're waiting for the microphone, can I just perhaps say the panel will do their very best to answer any questions, but cannot give specific advice about individual children and eligibility for specific trials. So just they may temper their answer with the limits of what they can answer within this environment. So, question from Carrie?
Yeah, my question is for Lynley. Forgive me, I probably, after all these years, I should probably know the answer to this question, but I don't, so I'm going to ask anyway. When you mentioned about SMPs, you talked about having a biopsy done at the point of relapse. So we've had a mixture of progression and relapse. At the point of progression, is it right to biopsy again then, or is there an assumption with progression that it's the same disease, just more of it? If that makes sense.
I think it's a very good question. I think we don't always know. I think it's right to consider biopsy. The answer may be no, but I think considering it at relapse, at progression, because it may be the same thing or it may be an evolution, and you may find, ALK is a good example. Why has a patient responding to an ALK inhibitor now got progressive disease which seemed to be controlled? What is the mechanism of resistance? Oh, look, the MAP kinase pathway has sprung into life and we could treat with a MEK inhibitor. So, of course, you've got to consider where is the disease, is it safe to biopsy, and why I think increasingly the ctDNA liquid biopsy studies are so important, because if you can get the same answer without an invasive biopsy, and possibly even a better answer, because that probably deals with tumour heterogeneity better. We know if you biopsy a tumour, you get a bit of a lump. What you're picking up in the blood, that tumour is shedding all bits of its DNA into the bloodstream. So it may actually be more helpful, and I'm sure Sally and others can comment on that. So very good question.
I did have a second one. Can I ask it while I've got the microphone? This one was mainly for you, Stephen. Thank you for your presentation. We've had treatment in America twice as part of our journey. We've never been MSK patients, and this might be an ignorant question, but it's a genuine question. Is there a reason why we haven't got some of the trial information from MSK in our presentations today? I ask because there is one that I was particularly interested in hearing about that I know is doing well in the US.
Yeah,
and
well, the copper sartrate, the copper sartrate trial is open at Memorial, and the GD2 SADA 16 and up trial is
this
open
was
there.
the RP1664.
No, I don't know that one. Yeah.
Okay, thank you.
Yeah.
Okay, so question. I think Leonie was next. So question from Leonie.
Yeah, thank you, and thank you, everyone. I'm just really struck by the point, and it's quite an obvious thing to ask, I think. I've been attending this conference in 2012, initially as a parent with my own interests, and now obviously supporting other families going through this. But from the presentations, it feels like there's a lot of trials, there's so many options, but then from a family perspective, we see that people run out of the path very quickly. And I know Helen Pearson has done so much work in how families make decisions about what to enroll on or not. And I'm just curious from a clinician point of view, how you manage that. When you speak with your parents and families, are you already thinking this is the best possible trial for this child at this moment, and then there may be X, Y, or Z after, or how does that work for you?
Okay, who would like? Steve, would you like to take that first?
Yeah, I can start. I mean, I think a lot of this is individualized decision making, where the role of the pediatric oncologist is providing options and providing information so that patients and families can make an informed decision. I try to counsel families to think about, particularly for children with multiply recurrent disease, to think about parameters, because you're right, there are a lot of options both at a given center, but for families who are willing to travel, then the options become more extensive. So we talk a lot about, well, would you want a clinical trial that includes chemotherapy, or are we only focusing on novel targeted therapies? Would you want a therapy that would require a hospital stay? Would you travel? So often it's a multiple step conversation of, there's been a relapse, these are the available options here, help me to understand what some of your maybe some of your limitations on thinking about options, and then it's my job to sort of go back, call some friends, and think about what might be available at any given time for a patient.
Yeah, I completely agree with that. And, I always, when speaking to patients and families, very much say that, I promise them that we will try to do the very best for their individual child and situation. The trials are the vehicles that we can use to get earlier access than we would otherwise get for many of these agents. But we're not serving the trials themselves, and that's really important. And a very wise mentor taught me to always say, you sign consent today. If you change your mind by the time you leave the building, in a week's time, in a month's time, we'll look at other options. We're here to facilitate helping you make decision, but it will always come down to what works for your family, your situation. The other thing is that although we have these long lists of options, we always have more patients that need them than the list of trials that we have, because not every option is right, or not every patient is eligible for all of those studies. We're trying to avoid duplication, but I've never had too many options available.
Okay, I think the lady in the striped top, almost at the back, was next.
Yeah, I was just going to say, there's a follow-on question there from that, if you want to ask that. Well, I was just going to make a comment more than a question, which is some of it is... I think that other piece, I agree completely with Steve and Lynley, but I see a lot of patients at the Children's Hospital of Philadelphia. We do a little neuroblastoma there. But some of it has to do with timing. A lot of it has to do with timing. Like Lynley gave this beautiful long list of trials, and Steve gave these categories of trials, but remember, these are very small trials. They're open for a period of time, and then they're closed to evaluate the safety of the patients that have already enrolled. So yes, there can be this very long list, but if you need a trial in a certain time period, they may be in a holding pattern. And or
so,
you're eligible.
for sure, that's before you even get to the eligibility issues. So some of it is timing, some of it is what do you want, what do you not want, what's appropriate for your history. But honestly, in real life, a lot of it is timing, and then it's the eligibility. So I have patients come to me having printed out all kinds of stuff from the internet, and then, copper sartate, great idea, very different. It's closed right now, so, sorry. I wanted to explain the logistics. Thanks, Ro. I think Nick had a comment to make following on from that.
I did. Is it working?
Yep.
I think it's difficult. Obviously, we heard from Lynley and Steve and Ro, and these people work in centres of excellence, and they're neuroblastoma experts, and clearly we invite them to this conference to speak to everybody for that reason. But it's not every child with neuroblastoma in the UK, let alone across Europe and North America, is treated in a centre of excellence by a neuroblastoma expert. And so, it comes back to the question of, if you take the extreme of innovation at the moment with cellular therapies, which is very specific in centres of excellence, but the ability to offer innovation through clinical trials in an equitable way with geographical spread, which is indifferent to socioeconomic factors, etc. It's a goal, but I think everybody is a long way from achieving that aim at the moment. And even the people in this room are probably different from many patients and families with children with neuroblastoma. So I think it's a very complicated topic, but we also have to think about the people that aren't in this room and the people that would never be able to travel to clinical trials, and how we give the opportunities to those children and families to have the same as, like myself, who was treated at the Royal Marsden.
I think Stuart has a
Yeah, if I
question.
could just make a comment to follow up on that. So for those that don't know me, I'm a bereaved parent. My daughter, Jossie, passed away 5 years ago. The reason I'm on this panel is because I have, lived experience of, going through clinical trials in a relapse setting. In some respects, this topic is really complicated. You see all of those different trials. All the science is very difficult to get your head around as a parent. But in other ways, it's very simple. And ultimately, what parents are looking for is they're looking for curative intent, at least. And that's at odds in this setting because clinical, certainly stage 1 clinical trials by their very nature, are around safety and dose, but as a parent, you absolutely don't see it like that. You're looking for a cure for your parent as you go, for your child as you go through those things. It's simple in the respect that you're looking for reduced suffering for your child. Ultimately, you don't want your child to suffer either now through the treatment or in the future through late effects. And it's also simple in the fact, and it's not probably been mentioned enough so far today, is that you're looking for hope and support. Yeah, and a factor in our decision was hope and being able to speak to someone who gave us hope. Now, it's important people are not given false hope, but there's always room for hope, and there's always room for people to hope for you as a parent. And they were all factors in our decision. So if I was going to boil it down simply, given all the science, we said if we've got those 3 things, they were the 3 most important things in terms of us making decisions in that environment.
Thanks, Stuart. I think Donna has a question online.
Yes, we do have a question.
I don't think we can hear yet.
Hello, can you hear me now? Not yet. Oh, he's going to switch it. Okay, thank you.
Yeah.
All right, the person online wanted to ask about the minivan study, and especially given the interesting data that you shared, Steve, where that stands and what's next. And then there's another question about precision medicine for you, Lynley.
Okay, so maybe I'll take the minivan question first. So the minivan trial was a trial that combined MIBG therapy with dinutuximab beta and nivolumab, so anti-GD2 and an anti-PD-1 antibody, which ran in Southampton and UCLH in London in the UK and in Madison in the United States. And over the course of 4 or 5 years, we recruited 43 patients testing that combination. Our last patients were recruited in October, and we still have some patients, one who's here today, who are still on trial. We will have data very soon in the next few months that hopefully we'll be able to present at ANR. But what we know so far is it was generally very tolerable, and we did see some good responses, but we haven't yet got an analysis that enables us to say whether putting those 3 things together is better than giving any of them individually, because those agents, 2 of those agents at least, the MIBG and the anti-GD2, have benefit on their own in neuroblastoma, and what we want to see is whether there's more than that additive benefit. So we will hopefully have that soon, and we can't really decide the next steps on what we will do with that until we see whether we've got a signal of benefit from the trial, particularly because the MIBG element of it is so complex to deliver. We need to be absolutely sure that it's a scientifically sound, and we've got some clinical evidence to support rolling it out into a multi-centre randomised study, but we hope we'll have that data at some point in the next few months. And so we go to the precision. What was the
Yes.
was there a specific question about precision
Yes.
medicine?
Yes, there was. The question is, given the vast number of targets, the vast number of drugs, really, it's pretty mind-boggling, and the combinations that you were talking about, can you comment on what, since those are 3 steps, right? You got to find the target, then you got to get the drug, and then does the child get the drug, and do they have a response? Do you have any comment on overall how successful you feel this effort is? I know there's an awful lot of disparate data, but curious about your thoughts on that, Lynley.
It feels like you've got to throw out a lot of fishing lines to catch a good fish, right? But we have to be doing that, and I think we have to see the early kind of phase 1 bits for what they are. You hope there will be individual benefit. You hope it will inform the next steps. But for example, the eSmart study, the Determined study, they are not meant to be kind of pivotal trials. They are meant to be signal-seeking trials. Patients can and do have benefit on them. But if you see benefit for neuroblastoma patients in arm A or arm F or whatever of eSmart, the real test is then taking that combination into a neuroblastoma-only study, and I would suggest that Beacon and the tiers that are there in Beacon 2 are a good way to do that, because then you're not diluting that population. You get a real answer for the disease in question. So I think they are successful, but we have to understand what the purpose of each of those stages are. Benefit for patients is always the purpose, but it's, at what level is it going to inform the next kind of steps? And we have seen things coming through that pathway that are looking positive for neuroblastoma. And so I'm encouraged by it, but it can't be the only thing we do. We definitely won't solve all neuroblastoma just with precision medicine, and that's why the immunotherapy, the CAR-T, they will all have a very big role.
Thanks, Lynley. Steve, I don't know whether you wanted to add anything to that? Or?
Yeah, I mean, I agree very much with that, and I like the line about having to throw out a lot of lines to catch a fish, because I think we look for what I would consider high-value targets where we think there's a reasonable probability that if the tumor has an alteration, there's a high probability of response. But unfortunately, we don't see those high-value targets very commonly. Yeah.
And just to say, we don't just randomly pick the targets. There's a group of very expert people, and the kind of neuroblastoma NDD generation of kind of meetings, academic meetings, where people are looking at the preclinical evidence, looking at the in vitro, the in vivo, really early stage stuff from adult cancers, but also pediatric specific targets. Increasingly, that's where we think the money's going to be, right? Not just taking the targets from adults and seeing if we can make use of them in children. We want to really focus on the pediatric specific targets, and being increasingly able to do that will be very important, I think. I can see Andy's hands going up.
Okay. So I think the lady in stripes has been waiting patiently for a long time to me.
Hi there, thank you.
This is for you, Steve. It's Joe.
Hello?
Oh, great. Yeah, we can hear you.
Hello everyone, I'm Cassie Lever. I am the mother of Jocelyn, who sadly died back in 2019. I've just also recently joined Solving Kids Cancer's Development Board. My question relates to something which I don't think has been touched on in any detail today, and that's the role in AI in this, and sort of to understand what's happening, what discussions are taking place, perhaps either side of the pond, and what developments, if any, have been made from an artificial intelligence perspective, be it in terms of diagnostics, prognosis, or perhaps most interestingly, drug development and any of the sort of targeted personalised therapies.
Excellent, really excellent question, and I'm glad I'm not on the panel.
It's
Yeah. I'm also Stuart's wife as well, so he won't thank me either.
So,
Glad it wasn't for me.
let's make Steve take that one first, as Lynley went first last time.
Well, it's a great question. I mean, so much of what we're hearing about is AI this and AI that. I would say our field is in its infancy in thinking about applying AI approaches to diagnosis and treatment. Some of the work that we're working on in the Children's Oncology Group is really focused on digitizing scans and, allowing AI to sort of help us understand which patients are likely to do well and which patients are not likely to do well. There's also some efforts focused on digital pathology, where you can digitize the scans and put those through the, an AI algorithm. But I would say we're really very early on in that work.
Yeah, the other area where there may be potential is in compound screening, rather than having to manually screen, thousands and thousands of compound. Have AI assistants to be able to do that. I think there's a real place for that there to help speed things up.
Thanks, Lynley. So I think Carrie was next, and then Andy and Cormac.
This is mainly for the UK members of the panel. We haven't talked much about the role of the NHS in all of this. And I'm just interested in your thoughts in terms of how the NHS can perhaps either help or hinder sometimes with access to clinical trials, because that has certainly been our experience. We don't tick a box, and therefore, are restricted to what our options might be. And I just wondered, when we talk about these trials, surely the NHS plays a part in this as well. We talked about eligibility, age, all the other factors, but sometimes, in my experience, we have found that the NHS has been more of a barrier than a help. I'm just interested in your own experiences.
Go first, Lynley?
Yeah. So one of the things that, for many reasons, we have an excellent NHS, right? But it's not limitless in terms of funding. And I think where we differ from a lot of other countries is the amount of money that goes into childhood cancer research from government. There aren't necessarily the same government level programs. I know that if I'm asked to go and review grants and kind of programs for other countries, they have a head start because X amount of money is put in for that program, and companies can apply to it or people can apply to it, and you can get trials running. Again, I think, as Steve said, the advocate voice is so important in this, and SKC are leading a very important piece at the moment for government to kind of look at what the priorities are. Priorities around trial setup times, I think, is a big thing within the NHS because of that. It's stretched. So funding PI time to be able to have the time to develop the programs and do the research is a key thing, but also having the resource to move that trial forward at every stage of setup, right? We want to be faster. So following the O'Shaughnessy report, which was mainly aimed at commercial study setups, there's a huge piece of work going on also to improve academic-sponsored studies, and Pam Kearns, together with SKC, is leading a piece of work called Impact, where there's a place at the regulatory table. It's looking at, clinical trial pharmacists. It's looking at, the radiology reviews for studies, the R&D things. So definitely there's room for improvement. That's the context we're working in, so we have to try and improve it within that context. It's definitely not perfect, but I think there is scope to make progress there.
Can I come in on that?
Yeah.
I think you're right, Carrie, and I think there's the way the NHS is structured fundamentally. I think one of the things that we as parents don't fully understand is that most people that are involved in clinical research are people from the NHS, people like Lynley, etc., and others. They most often, research is done at weekends and in spare time. It's not a core part of somebody's role. So people are first and foremost full-time clinicians. They're not people, we don't have enough people who have enough time to have protected time to devote to work on clinical research. And I don't know how you fix that, but it's just one of several, system issues that we face. Relative to other countries, we in the UK have have difficulty with things like off-label drugs, for example. So access to innovative, novel drugs is really only available generally through early phase clinical trials. And so it's imperative to try and get those clinical trials up and running as quickly as possible. And as Lynley alluded to with the impact study that SKC has kind of been helping to move, but it's a multidisciplinary group led by Professor Cairns. The reasons why clinical trials are not opening as rapidly as possible across the whole country is multifaceted. It might be different in one part of the country than it is in another part of the country. And there is an opportunity at the moment with the NHS long-term plan, the potential cancer plan, and the cancer task force that's just been reformed in the UK. But it will take everybody pushing together as one voice to actually make tangible progress. And fundamentally, there won't be any real tangible progress unless there is central government money that's put behind something. So there's only so much that, the big charities can do, let alone smaller charities such as SKC. So, it will remain to be seen, with all the best will in the world, whether there's actually going to be some funding effort that will actually be able to make tangible improvements.
Thanks. Just can I make one comment on that about it's not also just the NHS limitations on running trials, but once you have a trial and you have a positive result from it, implementing that as an NHS treatment, and that can take too long afterwards. But one thing that is crucial to that is that we need to have really high quality clinical evidence that a drug works in order to be able to get it funded in the NHS. And so the need to do things in a randomised way, in a robust way, and maybe do that for longer than it seems necessary to really prove that the drug is effective, is because we know that we won't be able to get it funded and delivered to all children as a standard of care unless we have really good quality evidence. So who was next? I think Andy was next.
Yeah. Thank you very much. 2 points. Just to echo what Steve said, accessing drugs for children with neuroblastoma is definitely better than it was 30 years ago, but it still is enormously challenging. And I think, patient advocacy got a major, major role. The second point is really to take forward Stuart's thoughts. So 15 years ago, I was pleased with myself if I had 10 drugs that children with neuroblastoma could receive, early drugs they were eligible for. But in retrospect, I was totally wrong, because those 10 drugs didn't really give hope, if you're with me. Their chances of working was poor. So I think clearly what we need to do is have options of combinations which really offer hope, if you're with me. And that requires a lot of really clever work, a lot of preclinical work. But I think, we should be offering children with relapsed disease, the best we can do in terms of hope. And we shouldn't be offering them, new drugs that have a low probability of helping them. That's much easier said than done, but I think that's what we should be aiming for. And definitely I was wrong 15 years ago. And when Nick used to come to see me in my office, some of the options I mentioned to him were just, inappropriate. I apologise.
Thanks, Andy. So I think, Cormac, I'll come back to you because I think Donna has got the question online first.
Yes, this one's for you, Steve. The online participant wants to know, what about the problems with production of MIBG, and is that going to impact some of the future trials?
Yeah, thank you for that question. It's very worrying for us in North America at the moment, where our sole supplier of MIBG therapy is on a production halt while they're working through some of their production issues. I don't think that's going to impact our next randomized phase 2 trial that I shared for the NANT, because that has a few more steps before it's ready, and hopefully the production issues are solved by then. But for patients who need MIBG therapy, outside of a trial as part of a compassionate access program, it's a major problem. For the group, it really, I think, highlights the differences, and Mark and I were talking about this last night, the differences between a regular drug that can be produced and stored, like an antibody or a small molecule inhibitor or chemotherapy, that can be stored, and if there's a production delay, it's okay because you've got some reserve. But MIBG therapy and the other radiopharmaceuticals are produced for that dose is produced for that specific patient. So there's not like a back supply of MIBG that we can tap into. So it's a tremendous problem.
Okay, thank you. Thanks, Steve. Cormac?
Thank you very much. So a slightly different tack. Traditionally, we have grouped children with relapsed or refractory disease together, and that's the title of the talk. So I'd be very interested to hear the panel's thoughts on, should we continue to group children with relapsed and refractory disease together, or should we treat them as different cohorts with different approaches and different treatments?
Okay, maybe Nick would like to go first?
We should definitely treat them differently, as different patients with different, and the truth is, we should have been doing that 10 years ago. There you go. I mean, I remember asking that exact same question at our very first parent conference in 2012 to John Maris at the time, actually.
Does anybody on the panel disagree with Nick?
Well, I don't disagree. I just wish we were smarter. Right?
I've always wished that, Steve. No disrespect. As smart as you are.
They're clearly biologically different, right? And ultimately, if we were smarter, we would be treating them differently, right? Like one fundamentally has chemotherapy and sensitive disease. The other population, at some point had chemotherapy sensitivity and then, then a relapse event. So I think there's a lot of work ongoing to try to understand, what's happening biologically for those patients. Why are those patients refractory in the first place? And what levers can we pull in the drug development space that would be reasonable to pull to match that biology?
So if I could kind of come back, so maybe more detail. So I think when you're doing very early first-in-child studies and you want to get a signal, you want to ensure a drug is safe, then having both populations together enables you to get to that end point as most rapidly as possible to recruit. I think the paradigm has shifted, and actually I think we've observed this with Minivan is probably the first study, where actually patients who were enrolled in high risk or who started up frontline treatment and then their disease didn't respond sufficiently for them to move to high dose, parents had the option to enroll them on the Minivan study, and then those patients who've had a sufficient response after going on Minivan have actually been able to come back up to frontline, have high dose, which enables them to have immunotherapy. And I think that's been the first kind of real shift and I think that's the paradigm to go forward, that you always want to try and keep patients moving forward on frontline if you can. And then with relapse, I mean, for all the issues that we do have in the UK, and the move towards having platform trials with Beacon 2, once that's fully up and running and established, it should be much quicker and cheaper and easier to add extra things into Beacon 2 once the infrastructure is established. So, you're reusing something instead of using something once, throwing it away, starting again from scratch. There are some of our European neighbours whose competent authorities won't allow them to run platform studies. So, you know, we definitely have challenges, but, you know, people are trying to work smarter and to reuse infrastructure and to make the money that is available go as far as possible.
So, Ro, I think, had a question?
I think, could I just make a quick
so
point?
yes, so Stuart first, sorry.
It's 2 points, and they're quite simple points, but I think the first thing for me is if you group those two things together, you've got the potential to mask significantly unmet need, because you've got everything grouped together in one place. I have no idea, out of all of the phase 1 clinical trials we've just seen, how many are focused on refractory and how many are focused on relapse, or whether we've thought about both of those things at the same time. So you need that focus, and separating those two groups is really important. The other thing for me as a parent is if you don't look at them separately, there's the potential for additional suffering for children, because you're giving them potentially therapy which isn't appropriate for that situation. So just those two things together, for me, you know, these things should be absolutely separated.
Yeah.
Can I just make a comment? I guess I would, not to be argumentative, but I would maybe take a little bit more nuanced approach and ask refractory to what? Because our assumption is that refractory means refractory to chemotherapy. But I think as we move immunotherapy to the frontline setting, we have to ask, what is it that you received and what has failed your child so far? As opposed to just line in the sand, you have relapsed disease, you have refractory disease. I think the world is changing and we need to change with it. And I think once we finally have optimal biomarkers for immunotherapy and other things that we're doing, I suspect that those biomarkers may be more important than our clinical sort of crude way of thinking about relapsed versus refractory. So I think based on the Beacon results and based on what the parents on the stage are saying, it's a reasonable thing to think about right now based on the data, but I guess I want a little more nuance and flexibility, as you expected.
Great, thanks, Ro. I think Leona has a question next.
Thank you. Yes, I just was wondering about children who relapse or have refractory disease who get back into an NED status, and then what happens? Is there more activity in this space now? Does there need to be more activity? What are the thoughts of the panel on what to do then, which is what we all hope for?
So I think Steve has some comments.
We for sure need more activity in that space, because with chemoimmunotherapy, we are now able to get some children back into a second remission, and there's very few trials in that space. I think because it's challenging to know if as Juliet said this morning, you only know if the agent isn't working, right? Because there's no disease to follow. So I think from a drug development standpoint, it poses some challenges. I think the only trial I know of for second remission in the U.S. right now is the Sloan vaccine. I'm not aware. I'll look at my friends. I'm not aware of another second remission trial in the U.S. I don't know what you've got here. Yeah.
I think it's definitely a space that
Yeah, I agree. And we've talked about whether we could have a maintenance platform trial for just those patients who've had a good response to Beacon 2 or other treatments, and then we're left with what we know there's still likely to be a high chance of further relapse without more treatment, and to whether that. But it is a tricky trial to design. I think Lynley has a comment.
I was just going to say that it does leave a space, though, to bring in novel designs of things like early detection of relapse, and think about, off-treatment liquid biopsy monitoring. If we can detect relapse earlier, would it make a difference if we can intervene early, or are we just subjecting people to unnecessary testing and actually it doesn't make a difference if you know earlier? Instinctively, you feel it must make a difference if you know earlier and can intervene more quickly, and there is work going on in that sphere. I think that's the kind of space where there's an option to look at that.
Yeah. Question from Lee?
Yeah, sorry, I couldn't not reply. So I think we will talk about this later, but I think if you would know indeed that there is a signal popping up and that what you're doing is actually not working, or there's a clone growing out or whatever, then, and most drugs have toxicities, so then I think we all would agree that it's not wise to go on with something you know it's not working. Yeah. So of course we will investigate this and we will find this out, and Mona Lisa will be one of the studies that will do so. But I think we all know, can state the obvious here, that that it might not be working. We might need to think of other ways to proceed.
Yeah, I completely agree. I meant in the off-treatment phase, when you're not actually getting any treatment, that's the option where we can see the value of that and test the value of that.
Thanks. I think Nick has a comment.
I've got a question. I guess when just thinking about what Rose said about refractory patients to immunotherapy or chemotherapy, etc. Back in my day, as a parent, you tend to see your child in a certain situation, and you look at children that are similar to your child but are further ahead, and you kind of try and navigate the future. And obviously, back in those days, there were children who had refractory disease at the end of therapy, and they were just treated ad infinitum on many, many, many trials, and probably it ended up worse because they were over-treated. And obviously, over time, children have become known who are seemingly refractory, don't clear their disease, and yet, for whatever reason, don't go on to progression or become longer-term survivors. So this is more a question from me to the clinical community about how to address that refractory population, and what are we doing about those patients at the end of therapy.
To leave? No.
I think this also boils down to the MIBG spots that end of treatment are still there, where we as clinicians sometimes say, okay, this it will fade away. It's like mature disease which will mature even more, and then it will become MIBG non-avid. And I think one way, so we published a series of patients with this phenotype, and what we said, so some have been biopsied, and it all turned out in those spots to be ganglioneuroma or to become ganglioneuroma. And I think one way forward is to make MRIs with diffusion-weighted imaging, because on an MRI we can distinguish ganglioneuroma from something else. So of course we have to learn here, but well, we did a small test. We haven't published it in our center, but we think we can identify these. So these are the patients we think are refractory. We might just have like mature disease.
Hi, I'm Meredith Irwin. I'll talk about biology later from Toronto. But like Leave, in Toronto, we published a series of these patients as well. And I do think MRI and maybe PET in certain circumstances can help. I think ctDNA will be a game changer for those patients. But I think this is another opportunity to collaborate, because I sense there's some biomarkers potentially, but there's so few patients. But we have many now, even 10-year survivors. But if you just look at their bones even on an MRI or a plain film, it's quite scary still sometimes. So it is hard, to your point, Nick. One of them developed secondary MDS, probably from all the treatments that we've done. So I think we do have to figure out those biomarkers as well, to figure out who needs more treatment and who maybe we should sit and wait. And I think ctDNA might give us a little more security with the watch and wait approach than just, MIBGs every few months.
Thanks, Meredith. So the gentleman with the check shirt behind you, just over there, has been waiting for a long time.
Hi. My name is Lawrence. I'm a parent. My son, also Lawrence, he's finishing frontline treatment next Saturday. So hopefully he doesn't ever move into the refractory relapse scenario. Just a question I have in general, from probably this morning's presentation and today, it seems to me that there's a huge improvement from, say, 25% survival rate 20 years ago to 50% or 60%. And I kind of got the impression that a lot of that improvement has come from immunotherapy type scenarios. And the common thing that comes up in a lot of presentations is this GD2 antigen, and how it's kind of harnessed in some way, whether that be in New York or CAR-T or monoclonal antibodies. So when you kind of if a lot of the improvement in the last 20 years has come from antibody type approaches, how do you kind of agree your trials across both the sides of the Atlantic? So you're working together and so on. So there's a lot of different trials which I get hard to understand. Some of the information, that's not my kind of area of expertise, right? So, but how do you decide what you're going to have a trial on? When you ask about advocacy, it'd be very hard for myself to advocate anything because there's so many different things going on, So, I think that's something that's kind of, and maybe that's just new to me, but I think that's very hard to advocate and say, I taught the CAR-T, then the presentation is very detailed. And that's, that's not a reason to advocate that particular topic, So I think it's kind of hard to understand. I think that the GD2 antigen activation, or whatever you want, adoption, looks to have been the one that has given the most improvement. So how do you kind of take that on board, or is all the improvement has been gained there, and do you move somewhere else? Because that's my kind of question. If it's
I think it's a really good question. So certainly within Europe, the CEOPAN trials, the adding anti-GD2 has probably added about 15% to survival. The other big step in improvement was probably the introduction of high-dose chemotherapy. But as the protocols get more complicated, it becomes really difficult to work out what the contribution of each element is. And when the high-dose chemotherapy trials were done, they added a certain percentage to survival, but that may change if that's followed by anti-GD2, because patients who benefited from high-dose chemotherapy may not also benefit from anti-GD2 and vice versa. I think Steve maybe has a comment about...
Yeah, thank you for the question. I mean, I think in the curve that Dr. Bagatelle showed this morning, showing the incremental gains over time, those early gains were from single transplant versus no transplant, and then 2 transplants, which is our standard in North America, compared to 1 transplant was another jump, and then of course the GD2 antibody was the last big jump. Your point about collaborating internationally, I think is really critical. We had a fantastic pre-meeting yesterday hosted by our SKC hosts, where we're talking through some of those very issues of what direction are we going next? How are we going to get that next big jump that we all really want? So it's we're really, I think, privileged in this field that we all work very closely together. We all work really well together and all have the same goal in mind.
Maybe Nick may comment on that.
Yeah, I think, I was talking as a parent, so I didn't introduce myself, Eddie. So if anybody doesn't know, I'm Nick Bird, who's a parent to Adam, and also I'm Chair of Trustees of Solving Kids Cancer UK, so remiss of me not to introduce myself. So going right back to Ro's initial talk about neuroblastoma, and it's one disease, but now, a form of precision medicine. Now there's low, intermediate, and within intermediate risk there are multiple different groups of intermediate risk and high risk. I think traditionally high risk disease has been treated uniformly, despite the fact that everybody knows that there are very different groups of children within the high risk category. And so if you look at GD2, at the moment, nobody's figured out which patients are most likely to benefit from having a GD2-directed therapy and which aren't. And that's one of the ongoing, pieces of research in the field to actually say, for all children, can we figure out who we should give this therapy to because there's the greatest chance to benefit, and who we should give other therapy to. And then in terms of things like targeted therapies, there are ALK inhibitors that are now coming into frontline, which a proportion of children may be able to benefit from. And I think the kind of biology has moved further forward than the drug development. So there are also other things implicated in the development and driving neuroblastoma that as yet cannot be targeted clinically. But as we move forward over the next 5 to 10 years, hopefully more of those things will be brought in as well. So that's just my personal perspective.
Thanks, Nick. And Stuart?
If I could just pick up on your point on advocacy, because I think it's a really important one, because I think, Steve, you raised the point, we need more parent advocates or patient advocates, and it is really difficult to understand as a parent how you can do that, because just as you say, we don't understand the science to the same degree as some of the experts we've got here. So, how do I contribute? And the way I see that is not kind of advocating for one treatment over another, because it's impossible to do that and you need to let the evidence speak to it. But for me, it's about, identifying where that unmet need is. It's about helping in terms of the setup of those clinical trials so that they work for parents in the right way in terms of the reality for the patient and the parents going through that process. And it's also building those coalitions. So it's obviously great we're all here today. The reality is for parents, or certainly my personal experience is, the environment feels very fragmented, if I'm honest. What's going on in the US, what's going on in Europe, and that in part is because of the rarity of the disease. In part it's because of pediatric oncology itself, but building those coalitions is so important as well. And I think parents have a really strong role to play in terms of making sure the right conversations take place internationally.
And I'll just add, we're stronger together, right? Like if I go to a company or to a legislative body alone, that's going to be less impactful than if I bring these guys with me, right? So I think we have to all work together to affect change.
We have a question at the back.
Hello, my name is Lynley. I'm Maya's mom, and Maya's currently undergoing the minivan trial. I believe she's probably one of the last candidates for them. Thank you so much for that. Maya, just bear with me. The question ultimately I hope is relevant. I won't be surprised if no one can answer the questions. Maya started, hello? Can you hear me? So Maya was diagnosed with high risk in April 2017. She went through rapid Kojak, frontline treatment, 4 years and 10 months in remission. We went through Solvi Kids Cancer for GD2, bivalent vaccine, MSKCC. So she was in remission for nearly 3.5 years. Then she relapsed. Then she had a treatment using the Beacon treatment, but not under the clinical trial, and she achieved remission again. At the time of remission in April 2024, there wasn't any trial, so it's just a comment on the doctor's comment earlier that sometimes it's the timing and also eligibility. In April, she achieved second remission. In July, she relapsed again, which kind of made her eligible for some trial. But at the time, minivan was closed. So we were going to go for CAR T-cell in Gosh. And before we went to that, minivan slot opened. So it's all about the timing. So just to tell you that sometimes it's the timing and eligibility. Now, the second point that I would like to make is I still have to find a like-for-like child neuroblastoma. So although it's nice to be in the same group of parents with neuroblastoma, I still have to find Maya's neuroblastoma is the same as someone's neuroblastoma. So blindly, I'll just say, if that minivan trial didn't work for the child, I can't just say that doesn't work, because that's not how neuroblastoma work. So from a parent perspective, I work as a specialist in diabetes. When we initiate patients with novel treatments, it's always about evidence care. 8,000 patients candidate was on this trial. And then here we are, I'm hearing minivan, 43 children in comparison to 8,000, you know, on SGLT2 or Ozempic or Mounjaro. My question ultimately is, because neuroblastoma is apparent now, you know, from this today's symposium, is very, very different. Is there anything happening, like a collaboration which was mentioned from the panel, that we could enroll more candidates from the likes of the Philippines? I'm originally from the Philippines, and I just feel lucky that we are under NHS and we've got access to the treatments. But for some patients that haven't got what we've got here now, and we should be really, really grateful, are we collaborating with other families that could potentially contribute to the trial, because we still have a lot to learn about neuroblastoma? And are we missing out on those data that are in existence in, for example, Indonesia, Philippines, Malaysia, or Turkey? Is there any collaboration happening, or essentially it is down to the cost, funding, or just lucky dip? If you got British passport or European passport or American passport, you're lucky, or Canadian passport. But if you come from a third world country, you have to sell all your, you know, finances and cows and carabaos and houses and estates before you can access something. I don't expect any answers right now, but I just want to hear if are we missing out on those information from those children, because essentially they are really might potentially contribute to the answers that we're looking for, which, you know, I believe, like what Nick Bird and Stuart Lever was saying, for us parents, we just want hope, we just want cure. We don't, you know, all these numbers, GD2, GD3, and all the numbers are letters, digits, they sound gobbledygook to me. Ultimately, I just want, hopefully the minivan is a ticket for Maya, but essentially we also want for other children, for the future, that we finally will find answers or cure that will be definitive for each child's cases. Sorry, that was long-winded.
No, thank you, Delaney. That's a really good point that I think a lot of people would agree with. I could perhaps just say that there is collaboration with lots of countries. Sometimes those countries you mentioned, it's limited by the clinical trial infrastructure in those countries to be able to undertake a trial that can deliver it to the standard that we need to for the UK and European regulatory authorities. And sometimes it's about cost. I don't know whether anybody else has comments to...
Yeah, I mean, I just, you know, direct you to the St. Jude Global program, which, you know, has largely focused on working with low and middle income countries to establish standards of care for, you know, for common pediatric cancers. That's been the first wave, you know, rather than bringing in novel therapies into those low and middle income countries, right or wrong, but that's been the approach that I'm aware of. Gudrun, I think maybe has a comment related to that. Yeah.
Maybe maybe the last question, because then it's time for us to have tea.
It was a response, I think.
Okay, fine. Yeah.
Sorry, I wanted to just thank you for this important comment and just add on to this, because I think there are several layers to this. Of course, the importance, of course, of opening trials in specific structures, all the regulatory issues, but also the important aspect that you mentioned of data and learning from each child and how the trajectory of each child can contribute to the future. And there are some initiatives, and I think we need to expand on them, on collecting data from children even treated outside trials. I would like to mention the SASHA International Initiative, which is just one example, led by Pablo Berlanga from Gustave Roussy, which aims at collecting data of children receiving off-label drugs. And I think such initiatives are hugely important. And if we can link them to data collection patients also about biology, etc. I think it will be a way forward, integrating also the other keyword that was mentioned, AI aspects. The more data we have, the more I think we can learn on how to unravel all the complexities.
Thank you, Gudrun. So one final question.
Hi, I'm Alex. I am a SKC trustee and mum to Archie. Archie passed away in 2018 and he had a really rough ride at induction chemo. He was in induction chemo in hospital for the full 4 months, was in intensive care twice as a result of the toxicity of treatment. And I think we've heard a lot today about how the improvements we've seen in survival is as a result of introducing more therapy and more treatment burden. And I was just interested to see how a lot of these trials, you talk about combination therapies and how that's better than looking at individual therapies. And I was just wondering, in an environment when we want to try and look at reducing the burden of therapies and the toxicity on treatment, why it is that we're looking at combining things rather than looking at things individually. And if we keep adding treatment, how do we start to step back from that?
Thanks, Alice. Maybe Steve?
Yeah, no, thank you for the question. I think the push to study agents in combination in the relapse population is so that we understand to what extent those novel agents may be candidates to advance to frontline, either as an add-on therapy, depending on the tolerability, or as a replacement, right? And so I think from my perspective, that's trying to really get the most promising agents into the front line as quickly as possible. If we spend too much time on monotherapy drug development, where my own bias is that the probability of benefit is lower, we'll just be that much further behind in terms of getting a new agent when we see success in the relapse space, getting that to the frontline space.
Last comment to Nick.
Oh, you've always been my favourite, Juliet. Thanks. Last comment.
I was your favourite.
Yeah, you're not anymore. I completely agree, Alex. And there's always the law of unintended consequences, right? My favourite phrase, people that know me. And obviously behind that curve, as you lift that curve up, generally speaking, that curve has been lifted up by giving the children on the curve below an extra therapy that they didn't need. So there's always this cost trade-off that you've improved survival by giving those children who would have survived anyway something that they didn't need. And then there's this massive fear that if you remove anything, you're going to do worse. And now children who would otherwise have survived are no longer going to survive. And I think the only real way out of that is for all of these super smart people to be even smarter and to figure out which of the children need which of the therapy and which of the children need some different type of therapy. And so, I don't think there's going to be obviously John and Francesca, they're optimists with respect to CAR T-cell therapy, etc. But I think, I sat in a room in 2010 probably, and it was neuroblastoma treatment is it's the most intense treatment of all, and we've made it as intense as we possibly can. And then that was before GD2 was added on in Europe, and it was before tandem transplant. And here we are. I think there now is genuinely a view that you just can't keep adding and adding and adding for every single patient.
Thanks, Nick. So I would like to end by thanking our 4 panellists and everybody in the audience who put such insightful questions to them, and for the really lively discussion we've had. And I will just hand back over to Cormac to give us our instructions for tea.
Thank you very much. Thanks, everybody, for a great session. So that concludes the session at the minute. And we'll be back at 4 p.m. GMT to discuss measuring the response to treatment.